Por tanto, si a Dani Guinar le pedíamos algo difícil, pues a Silvio, imaginaros lo mismo, ¿no? Danos en pocos minutos unas pinceladas de qué hay de los nuevos tratamientos en enfermedad inflamatoria intestinal. Eso podría ser una semana a 24 horas cada día, pero seguro que Silvio será capaz de resumirlo en pocos minutos y dar información súper útil para todos los que nos estáis escuchando. Silvio, cuando quieras, adelante.
Hola Miquel, and felicitaciones por el nuevo centro de inflamación sistémica con or autoimmune diseases or inflammation center. I think that this is great news. So, dear colleagues, in the next few minutes I will give you an overview about the new drugs and the future of IBD treatment, and this is a real pleasure. Thank you so much for the kind invitation. I think that these have been great years for our patients with inflammatory bowel disease, particularly when we look at the overall drug pipeline that we have been developing for patients with Crohn's disease and ulcerative colitis. You see a lot of green tackling the different mechanism of action that sustain intestinal inflammation. And the maturation of the last phase 3 has really led to a lot of therapeutic options for our patients. And in fact, I will discuss particularly on the success and on how the field is moving, particularly with all the pathways that you see here on the leukocyte cell adhesion, S1P modulators, IL-23 blockade, and the several JAK inhibitors that we have in our clinical armamentarium. So let's start with anti-cell adhesion molecules, and selectivity has been really the driver for success, and in fact blocking alpha-4 beta-7 has been one of the key therapeutic targets for vedolizumab, and actually there is only one drug left in this area. All the others, as you know, have been tested but not successful as vedolizumab. The next step is to go oral, and this is the Morphic compound, oral blocker for alpha-4 beta-7, successful reduction of histological inflammation in patients with ulcerative colitis. We will see whether this is going to be developed and read out soon successfully for the phase 2b that has been just completed. We'll see the results very soon. So let's now look at still leukocyte recruitment and how traffic control can be inhibited, not only from the arrival, so anti-alpha-4 beta-7, anti-integrin, but particularly for what happened to leukocytes from the gut, migrating through the lymphatics, through the lymph node, and then their egress, where the immune inflammation is primed. There is a gradient that regulates the exit of leukocytes from the lymph nodes towards the periphery. And this gradient is dominated by sphingosine 1-phosphate and the receptors expressed on leukocytes. And now we have cells that are able to block the leukocytes inside the lymph nodes, avoiding the egress towards the periphery, and therefore leading to less inflammation to the tissue where they are supposed to give the damage. In this case is the intestine. And these are the two drugs that are mainly acting as modulator of S1P, etrasimod and ozanimod. Just as a reminder, these receptors are expressed very widely, and the selectivity may lead to a better profile. The first one is ozanimod. These are the registration trial that we have conducted now, I would say 4 or 5 years ago. And when you look at all the bars for induction on the left side in blue for ozanimod versus placebo, this is ulcerative colitis, and also for maintenance, you see that everything is successful, suggesting that ozanimod is a great drug for patients with ulcerative colitis. Next generation is etrasimod, and this is also the latest approval of S1P modulator. This is already available in clinical practice for multiple European countries. You see that both induction and maintenance endpoints have been met in patients with ulcerative colitis. Whether these drugs will be developed also for Crohn's disease is unknown. As you know, ozanimod was a failure for the first induction study. We need to see all the other reports later on during the year. Anti-cytokines. Anti-cytokines after anti-TNF, we have all the big arena of the IL-23 inhibitors. Let's start in understanding what is the evolution of IL-23 inhibition. The first was ustekinumab, blocking both at the same time IL-12 and IL-23 simply because they share P40. But when you look at all the next comers, they block uniquely and selectively IL-23, and this leads to a better effect. And in fact, in Crohn's disease, all the trials are successful. This is the Lancet registration trial for risankizumab in Crohn's disease. You see different induction doses, very effective as compared to placebo for induction and also for maintenance. So no doubt that risankizumab is a great drug. Actually, it's even more powerful than the first generation ustekinumab. This is the sequence was just published. We published this in the New England a few months ago, and when you look at the efficacy, you see that there is superiority at 1 year, not only for symptoms but also for endoscopy. Second drug in the class, mirikizumab. In this case, a very interesting and effective drug as well as risank, but superiority was not met versus ustekinumab. This is actually interesting because it leads to the fact that not all the IL-23 are the same, but this is of course actually was published a couple of weeks ago in the Lancet. And then you see these are the results, as I mentioned, of non-superiority versus ustekinumab. The third and the last one is guselkumab. Guselkumab very effective in Crohn's disease, not only for the induction and maintenance as you see here in this slide on the left side. Actually, this is a very special study because it leads to the concept that you can induce and maintain, and you need to look at the overall picture of the patients according to treat to target. But also you see here, as compared to the purple, any endpoint shows superiority of guselkumab versus ustekinumab, so confirming that also guselkumab is a better drug as compared to the first generation ustekinumab. What about ulcerative colitis? Very similar. In ulcerative colitis, mirikizumab, highly effective. This is the New England registration trial. We have this drug already approved and using it. We have treated a lot of patients in our center, for instance. This is guselkumab. Once again, you see nice induction data and also maintenance data. So this is a drug that we are waiting to get approved, but actually this is very interesting. And the third one, risankizumab, also for ulcerative colitis effective. So all the promises of the IL-23 have been kept. The real issue that I see, and this is you see all the endpoints for risankizumab for the maintenance. The real issue that I see is how we are going to choose what is the future for this drug. The future is going oral. Oral actually could be even better, but the major problem is how to choose having so many drugs that inhibit the same pathway, because they look very similar in terms of efficacy, and this is just a big question. Now let's move towards from cytokine as monoclonal to cytokines intracellular. All the JAK inhibitors are great drugs. We have 3 available in our practice. You know that JAKs control multiple cytokines, so this is a true combination treatment, and this is the first one, filgotinib. This is the registration Lancet trial, induction and maintenance, very effective in both bio-naive and bio-exposed patients. The second one, considered one of the most effective, is upadacitinib. This is the Lancet for induction. Not only in the induction but also in the maintenance. Half of the patients will do well, so this is quite effective molecule. And what is interesting is that this drug works very well also in Crohn's disease, as you see in the registration trial, not only for the bio-naive but also for the bio-exposed patients. So I think that JAK inhibitors are really great advances. And finally, something that is very interesting for our clinical practice is the speed of action. How fast are these drugs and how effectively inflammation can be kept under control. And even in network meta-analysis, you see that TUPA is considered, at least in ulcerative colitis, one of the best and fastest and effective drug that we have in our therapeutic armamentarium, as recently reported in this network meta-analysis. So you have seen a huge number of drugs. A lot are still under development, as you see here in this overall picture. The real issue for me is that when we look at the success and failure, well, 30-40%, maybe half of the patients are in full clinical steroid-free remission, but the major issue is that today we are not breaking the therapeutic ceiling. We need to take multiple approaches to go beyond these 30-40%. Multiple steps can be taken here, but I think the real future is going to be to changing our vision for one treatment fits all, one size fits all, to the personalization of treatment in understanding what is the pathophysiology of each patient and how this can lead to the future. For this, I think that we need to do a lot of research, and an example is the IPOP integrative personalized omics profile. There is a lot of research going on omics, on network medicine, understanding the controller of inflammation, and this is something that we are developing at multiple levels. One example is this innovative medicine initiative that awarded over 15 million in order to see whether we can detect the patients in ulcerative colitis that can respond to specific treatment. And with this, I really thank you very much for your attention, and I will be very happy to take any question. Thank you very much.